Calcium is the most abundant mineral element in the body. Extracellular calcium functions in bone mineralization, coagulation, and stabilization of plasma membranes. Intracellular calcium functions in muscle contraction, hormone secretion, mitosis, and glycogen metabolism. It is in a hormonally regulated homeostatic equilibrium with blood, bone, bowel, and the kidneys. Approximately 98% of the total body calcium is stored in the skeleton in the form of hypoxyapatite, and it is a lattice-like crystal composed of calcium, phosphorus, and hydroxide. The normal range for the total calcium in serum for an adult is 8.5 to 10.5 milligrams per deciliter, or 2.2 to 2.7 millimoles per liter. 40% of the total calcium in the blood is bound to plasma proteins, especially albumin. The remaining 60% is unbound and ultrafiltrable. It exists either complex to anions, such as phosphate, or as free ionized calcium, which is biologically active form. Factors that affect the relative concentration of each form of calcium in the blood include plasma protein concentration, acid-base abnormalities, and ion concentration. Factors that affect the total concentration of calcium in the blood include PTH, vitamin D, and to a lesser degree calcitonin and ulcerations. 67% of filtered load is reabsorbed in the early proximal convoluted tubule. 25% of the filtered load is reabsorbed in the thick ascending loop of Henle. 8% of the filtered load is reabsorbed in the distal convoluted tubule. The remaining is excreted. At the early proximal convoluted tubule, via primary active transport, the basolateral 3-sodium 2-potassium ATPase pump utilizes the energy of hydrolyzed ATP to create and maintain transmembrane electrochemical gradient. The gradient facilitates both secondary active transport by the luminal or apical sodium hydrogen antiporter, NHE3, of sodium into the cytosol, and the paracellular transport of calcium into the interstitial fluid respectively. At the thick ascending loop of Henle, via primary active transport, the luminal renal outer medullary potassium channel utilizes the energy of hydrolyzed ATP to expel potassium ions from the cell into the lumen, complementing the activity of the sodium-potassium ATPase pump. The electrochemical gradient facilitates both secondary active transport by the luminal sodium-potassium 2-chloride cotransporter, NKCC2, of sodium to the cytosol, and the cation-selective pore-forming claudin-mediated paracellular transport of calcium into the interstitial fluid, respectively. At the distal convoluted tubule, the electrochemical gradient facilitates secondary active transport of sodium into the cytosol via the luminal sodium chloride cotransporter, NCC, and the selective epithelial sodium channel, ENAC, as well as the transport of calcium into the cytosol via TRPV5. Next, the intracellular calcium concentration is buffered by calbidin-1. Finally, calcium exits the cell into the interstitial fluid by secondary active transport via the basolateral sodium-calcium antiporter, NCX, and by the primary active transport via the calcium ATPase channel, PMCA4. For physiology, pathophysiology, or pathology, be sure to like the video, subscribe, and follow us for more.